PENTRA C200 delivers a new milestone in clinical chemistry development by combining a high degree of automation, ergonomics and quality of results into one compact system. Everything was considered to ensure your routine work is made safer and easier. The PENTRA C200 is a discrete photometric benchtop chemistry analyzer for use in clinical laboratories. It is not intended for use in point-of-care settings. It duplicates manual analytical procedures by performing various steps such as pipetting, mixing, heating and measuring color intensity. The PENTRA C200 is intended for quantitative measurements of a variety of analytes: glucose, sodium, potassium, and chloride. ABX PENTRA Glucose HKCP reagent with associated calibrators and controls are for quantitative in vitro diagnostic determination of glucose in serum and plasma using glucose hexokinase method by colorimetry. Glucose measurements are used on the diagnosis and treatment of carbohydrate metabolism disorders including diabetes mellitus, neonatal hypoglycemia, and idiopathic hypoglycemia, and of pancreatic islet cell carcinoma. The ISE ion selective electrode module is intended for the quantitative determination of sodium, potassium and chloride in serum, plasma, and urine by potentiometry using ion selective electrode with associated reference solution, calibrators and controls. Measurement of sodium, potassium and chloride are used in diagnosis and treatment diseases involving electrolyte imbalance. The PENTRA C200 is a benchtop clinical chemistry analyzer using two measuring principles: absorbance and ion selective electrodes. The instrument may be summarized as follows: multi-parametric, up to 15 simultaneous tests plus 3 eyes tests, on routine or stat. 90 without eyes to 360 tests per hour, with eyes analytical cycle of 40 seconds, random access working on primary tubes or sample cups. Reagent cassettes are compact and ready to use. Onboard barcode readers are used to identify newly loaded reagent cassettes and samples for patient identification. Eyes module. PENTRA C200 includes an optional ion selective electrode module which contains a sodium electrode, a potassium electrode, a chloride electrode, a reference electrode, 2 calibrators and 2 controls. The direct application is intended for serum and plasma. Indirect application is intended for urine. Glucose reagent contains a bi-reagent cassette, ready to use, reagent 1 R1, and reagent 2 R2. R1 reagent contains NAD, ATP, buffer, and sodium azide. R2 contains hexokinase, G6PDH, magnesium sulfate,